Hello, everybody, and welcome to Technology Today for June 22nd, 2025. It's Season 2, Episode 25. I am MayorWatt. I run ohmTown.com, and along with the Sentient AI, who's not available today, we talk about the news. In this instance, it's 5 technology-based articles, technology-focused articles. Years before diagnosis, saving people from stroke. Let's talk about axolotl skin, gene jumping, and a nanotramp. Hopefully these aren't going to be too difficult deep dives, but I'm going to try and make this really quick. We're going to go in, talk about these articles. It's science and technology based, and then we're going to kind of jump on out of here. It is a time machine episode. I'm trying to play catch up because my mayoral duties kept me from doing the show on time, so I've never missed an episode yet in 3 years and 6 months now. So let's get this going. Yeah, everything's powered by ohmTown, so go and check it out. It's that site right there. All right, so that is ohmTown's front page for the 22nd, but that is the landing page for Technology Today over at ohmTown.com. And while it has some of the news, we do sample from other areas of the site. And I just realized that I did not put, I didn't prep all of the articles the way that I normally do. So anyway, if you want all of today's show notes, you can hit !TT over in Twitch, and Ohmatron will spit out the articles into the chat. That said, the first article is doctors find that they can detect cancer in blood years before diagnosis. I'm really curious about what they're detecting because based on my research, I could probably detect it, but all right. Anyway, so researchers at Johns Hopkins University have discovered that cancer can be detected in the bloodstream a full 3 years before it's spotted by doctors for an official diagnosis. That official diagnosis is typically the embodiment of cancer, that not just precursors to it or clues to it or even your blood chemistry indicators. I think that they are going to find free-floating cancer cells, CTCs. They basically are cancer cells that are floating in the blood. But let's find out. Now, I didn't prep the show like I normally do. It's detailed in a partially government-funded study published in the journal Cancer Discovery last month. The team found that genetic material being shed by cancer tumors, that's exactly what it is. Far earlier than previously thought, paving the way for promising new cancer screening methods that could potentially head off the disease long before it gets more serious. So when you click the link, it takes you over to the source. This is over at futurism.com in a section they call Neoscope. Victor Tengerman put the article together. 3 years earlier provides time for intervention, dramatically more time for intervention. It'll be very difficult to find the location of what it is that's shedding this genetic material, but let's see what they actually talk about. In some cases, the advanced detection could make the difference between being able to beat the cancer or not, according to researchers. 3 years prior. So, however, there's plenty of questions remain as to how useful early cancer screening tests can turn out to be in practice, and researchers will have to figure out how to turn a positive test result into potentially life-saving. So I'm really curious what they're saying. The researchers found that their multi-cancer early detection lab test correctly identified 8 as showing early signs of cancer. 4 of the 8 showed additional signs of tumor-derived mutations. The study shows the promise of MCED. Using advanced genetic sequencing techniques, they analyzed 26 blood samples of participants who had been diagnosed with cancer within 6 months and compared to them to 26 other samples of participants who weren't diagnosed with cancer. And they found those relationships there. 4 of the 8 showed additional signs of tumor-derived mutations, and the MCED lab test correctly identified 8 as showing early signs of cancer. So how did they actually do it, though? They don't actually talk about it. I'm going to have to hunt down this study. They just said the tumors are likely to be much less advanced and more likely to be curable, which is naturally. So I'm going to have to find the study. They actually link to it over in the article, but I don't read the article or the studies live. I guess we could, but that would take different planning, and it would be a much longer show to talk about this kind of stuff. But earlier the detection, obviously the better. And I'm really curious about what they are actually looking for. But they say that the lead author says 3 years earlier provides time for intervention. The lead author is at Johns Hopkins University School of Medicine, a professor of oncology, Yushuang Wang, in a statement that the tumors are likely to be much less advanced and more likely to be curable, but you're going to have to find them. Otherwise, you have to do chemo, and that is very, very destructive to the flora and fauna of the body. It basically kills everything in its path, in the hope that it's going to wipe out the tumor and other markers in the blood. It's just not a pleasant experience. If you've never witnessed anybody going through it, radiation therapy is one thing, chemo is another, and there's other treatments, but yeah. Anyway, let's keep on going. The next article is over in Non Sequitur News as well. Stanford doctors invent device that appears to be able to save tons of stroke patients before they die. Ischemic stroke from brainstem clotting, basically blow out a blood vessel. As Stanford explains in a blurb, the novel millispanner device may be able to save the lives of patients who experience ischemic stroke. Traditional clot removal, a process known as thrombectomy, generally uses a catheter that either vacuums up the blood blockage or uses a wire mesh to ensnare it. A procedure that is rough and imprecise as it sounds. Conventional thrombectomy or thrombectomy has a very low efficacy rate because of this imprecision, and the procedure can result in pieces of the clot breaking off. Correct. Again, when you click the link, you'll get taken over to the source. Oh, look, it's another futurism source. Over at Neoscope, Noor Al-Sabai put the article together. They say it's a sea change. I'm kind of curious what this is because it looks like a stent type device. It's a novel device that literally spins away the clots that block blood flow to the brain and cause stroke. So let's see. Oh, it's a spinner, not a spanner. I think it said millispinner. Also enters the brain with a catheter, but instead of using a normal vacuum device, it employs a spinning tube outfitted with fins and slits that can suck up the clot much more meticulously. Stanford neuroimaging expert Jeremy Haidt, H-E-I-T, who also co-authored a new paper about the device in the journal Nature, explains in the school's press release that the efficacy of the milli-spinner is unbelievable. For most cases, we're more than doubling the efficacy of current technology, and for the toughest clots, which we're only removing about 11% of the time with current devices, we're getting the artery open on the first try 90% of the time. It's a sea change technology that will drastically improve our ability to help people. Congratulations. So what's unique about the milli-spinner is that it applies compression and shear force to shrink the entire clot, dramatically reducing the volume without causing rupture. So I think it's twisting it so that it can actually tighten it up instead of going in and trying to grab it and pull it out. And like anything that is elastic, there's a shear point where it's just going to detach. You get a piece of it, but then there's other pieces that might be dislodged and go further in or actually cause a rupture. They say it works so well for a wide range of clot compositions and sizes, even for tough clots, which are impossible to treat with current technologies. Our milli-spinner can treat them using simple yet powerful mechanic concept to densify the fibrin network and shrink the clot. Yeah. So they literally just tighten it up. Think of taking a rubber band and twisting it, and it compresses more and more and more. As long as you don't do too much, you'll be able to retrieve the whole blood clot. All right, let's keep going though. The next article is over in the mobile channel. Axolotl skin mucus. Mucus. As a potential weapon against multi-resistant bacteria and cancer, axolotls are known for their ability to regrow limbs, organs, and even parts of their brain and heart. But the tailed amphibian with the scientific name Ambystoma mexicanum can do even more. Its mucous membrane protects it from pathogens, and more precisely, in antimicrobial peptides that are on it, and in it contains. This property makes the amphibian interesting in medicine, as the increasing resistance of bacteria to antibiotics is a major problem in the healthcare sector. Again, you click that link, it takes you over to the source. It's over at phys.org. I don't. But all right. Phys.org is where this is housed. Inca Burrow and Hanover Medical School put the article together. Lisa Locke and Robert Egan put the article together by editing and reviewing it. Axolotls are awesome. If it wasn't for the fact that I don't want to keep them out of nature, I would want one. Because they just look so cool. Anyway, and I'm working on a project, and somebody said, "Hey, can you put an axolotl on that?" And I'm like, "Well, yeah, sure. All you have to do is contact us, and can make it happen." So, anyway, more on that at some point if it becomes a reality worldwide. I don't know. We'll see. Researchers are therefore looking at for new target molecules that can replace the increasingly ineffective antibiotics, AMPs, or antimicrobial peptides, can replace the increasingly ineffective antibiotics. They're among the most promising candidates. They are part of an innate immune system and are found almost in all living organisms. The research team, led by Dr. Sarah Strauss, head of the Kirsten Reimers Laboratory for Regenerative Biology at the Clinic for Plastic, Aesthetic, Hand, and Reconstructive Surgery at Hanover Medical School, has studied various AMPs from the skin mucus of axolotls living in the laboratory. So these antimicrobial peptides could be an alternative to antibiotics in the future. They have a broad spectrum of activity, and at the same time, it is more difficult for pathogens to develop resistance. It's more natural, I suppose. It says axolotls were gently massaged with sterile gloves, and the mucus produced was removed from the gloves with sterile scrapers, all in accordance with the guidelines of the German Animal Welfare Act. So they find that its special chemical structure can contribute a resistance against the methicillin-resistant Staphylococcus aureus, or MRSA. Bacteria. Basically, MRSA is really horrible because once it takes hold, it's very difficult to get rid of. Oh my goodness. All right. I'm going to try and move on, but I have a wild animal inside the studio and it's going to cause problems here any second. Okay, let me keep going. The next article is over in Technology Today. Hold on. I need to fix something. All right. So jumping genes found hijacking cell division to rewrite human DNA. Line 1, a jumping gene making up 20% of the genome, invades DNA during cell division using protein RNA clusters, offering insights into genome evolution and disease. Viruses are masters of hijacking the cells they infect. So when you click this link, you get taken over to SciTech Daily. NYU Langone Health put the article together, and it says here that viruses are masters at hijacking the cells they infect. Over time, our bodies have learned to silence most of these foreign sequences, but not all of them are inactive. A few remain restless, earning the name of jumping genes because they can still move around the genome. Among them, one stands out, one called Line 1, short for Long Interspersed Nuclear Element 1, and it's the only one still capable of copying and pasting itself entirely on its own. Line 1 works in a clever way. It first creates a copy of itself using RNA, the close chemical cousin of DNA, then the RNA is converted back into DNA and inserted into a new spot in the genome. This copy and paste process is similar to how the retrovirus HIV operates, which is why line 1 is known as retrotransposon. In this way, it adds code to the human genome every time they move, which explains why 500,000 line 1 repeats now represent a staggering 20% of the human genome. These repeats drive genome evolution, but can also cause neurological diseases, cancer, and aging when line 1 randomly jumps into essential genes or triggers an immune response like a virus to cause inflammation. That's one of the things I don't know. I've asked other doctors, like, what does inflammation look like in this kind of context? And I've never really gotten a response that makes any sense. Nobody has explained it to me. What does inflammation in blood vessels look like? What does inflammation look like? But maybe it's just too basic, and the doctors don't really seem, or they don't. I don't know how to explain that. Maybe they just say, well, this is what it looks like, or this is what it is. But I'm really curious what inflammation looks in a body, and why it poses such a problem. So, led by researchers at NYU Langone Health and the Munich Gene Center at Ludwig Maximilians Universität in München in Germany, the work revealed specifically that line 1 can only bind to DNA when ORF1P, which can bind to RNA, DNA, and itself, is linked copies called multimers, accumulates into clusters of hundreds of molecules called condensates. As more ORF1P molecules build up, they eventually envelop the line 1 RNA, which makes more binding sites available for the entire cluster to attach to DNA. So it kind of becomes a hot mess of merging line 1 condensates. They have a remarkable feature in that their DNA binding ability emerges only when the ratio of ORF1P copies to RNA is high enough in the condensates. So moving forward, they'll be looking to see if other condensates undergo functional changes as the ratios between their components change. Quite intriguing. Okay, let's move on. Our last article is over in Technology Today. Physicists built a trampoline smaller than a human hair, and it could rewrite the rules of microchip design. The world's strangest trampoline doesn't bounce. It swings sideways and even glides around corners, but no one can jump on it because it's less than a millimeter tall. Imagine a trampoline that is so tiny, it's just 0.2 millimeters wide with a surface thinner than anything you've ever seen, only about 20 millionths of a millimeter thick. That's what's going on in this experiment. The article is over at SciTech Daily. University of Constance put the article together. And it says here, so why design the trampoline if nobody can jump on it? Of course, this construction was not designed for people to use. The brains behind the trampoline, physicists from the University of Constance and the University of Copenhagen and ETH Zurich, want to use it to demonstrate new methods of phonon transport. So phonons are, so to speak, sound quanta, i.e. the elementary excitations on which vibrations of the crystal lattice of a solid are based. Use the trampoline, the scientists or physicists want to demonstrate how phonons can be directed around corners by means of a unique surface structure based on mathematical topology principles with practically no loss of momentum. This is important, for example, in microchip circuits where signals are to be directed around edges and curves. You can actually shape the pathways in a different architecture. If you can actually send it around corners, the conduit can be a different shape. It doesn't have to be how they are now, in like 90-degree turns and stuff like that, or more graceful. It depends on what they're actually doing here. The amount of phonons that bounce back instead of going around the curve is less than 1 per 10,000. This ultra low loss is on par with contemporary telecommunications devices. They're interested in studying exactly these kinds of topological effects and surface structures and how they can be used in applications. And they think that with this method, it could be possible to build entire roads for phonons. They created a trampoline-specific design. His colleagues, their colleagues, from the University of Copenhagen and ETH Zurich then put the idea into action, and the research team's results were recently published in the journal Nature. There is more in this article, but essentially this technology has the ability to enhance circuit design, chip design, mainly because it offers the potential of being lower resistance, making it more efficient overall. And with greater efficiency is lower heat and data loss. But pretty cool. We're going to hop on our trampoline, and that's going to send us back to the front page of Technology Today over at ohmTown.com. Go over and check it out. Become a citizen, sign up or sign in, read all of the news, all news, because all the noise comes from me. And I'm going to get out of here. We're going to reset real quick and do FourWheelTech. We'll see you in a little bit. Bye-bye.